morning. Thank you all for waking up early to hear what we have to say. I'm glad to be here. I'm glad to kind of welcome you to Barcelona, even though I'm transitioning, as Beppe mentioned. So I'm not fully there, but I'm kind of shifting over. I'm also glad that the first talk gave you a flavor of the importance of clinical translation. And I think I will give you the tip of the iceberg of the kind of work we've done in the last 10 years with what I'm sure Fran will call a weird material, which is called graphene. So those of you in the business of nanomaterials and nanomedicine and the use of nanomaterials for medical applications, the overall aim, I think, is summarized here. We're all aiming to transform nanomaterials for next-generation medical technologies, whether these are, as you've heard, drug delivery or biosensing or devices or tissue engineering, depends on where you are. And I think we're going to hear about all of these during those next 3 days. But the ultimate aim, if you want my opinion, having done this work for almost 30 years now, is that we're all judged at this bottom line. How do we take the materials to the clinic? And when we started the program in graphene and 2D materials, 11, 12 years ago now, that was one of the key elements and the key questions we've been asked. So I'll take you back almost exactly 10 years ago, a bit over 10 years ago now, when we were building the program with Kostas, the Nobel Prize winner in graphene, and we were landscaping in this paper that I don't know if you read it or not, but it's just giving you a flavor of where does graphene back in 2013 sits in relation to the other two nano forms of carbon nanomaterials, fullerenes and carbon nanotubes. And you may identify already that there are plenty of possibilities in this column in relation to these other two columns. However, the reality of the situation back then was that there was absolutely no in vivo study in the literature, and I'm not talking about human, I'm not talking about large animal studies, I'm talking about little mice studies, nothing. There was absolutely zero. So when we were kind of tasked to build a clinical program around graphene, the challenge, that was in 2015, was kind of summarized, distilled in these bullet points. First, we had to envision and design a clinical translation program around graphene materials. Remember, clinical translation program, not preclinical. The second was that the funding agencies, both in the European Commission and in the UK, were generous and told us, you can get money for the next 5 to 7 years, but you'll need to deliver and get graphene to the clinic, if you can. And by the way, we're not telling you that COVID is going to hit. So that was the challenge, that was the task. So what I'm going to do in the next few minutes is to give you two separate but in parallel clinical translation programs we've run. As I mentioned to you, I'm just going to give you, because of the interest of time, the tip of the iceberg type of effort. So I won't be able to tell you everything that went on in the last 10 years, but I will try to give you where we landed and where we are right now. The first thing we did was to establish the material. Remember, this material was very new. So even now, when we talk about graphene, I'm sure that if I do a survey here in that room, there will be at least 10 different interpretations of what graphene actually is. There's the prototypical form, and there is these other different forms of functionalized surfaces, surface functionalization, or double stacking, triple stacking, all kinds of different. The bottom line is that we're talking about a single sheet of carbon atoms. And when we were looking to design a clinical program, we wanted, of course, to use a material that would be basically water suspendable, because we knew that all of the biological and physiologically relevant tissue and material and biofluids will be water-based. So If you run, again, a survey back then, as we did, I believe there's a paper in Nanoscale in 2015 that we published where we surveyed the whole literature and identified which are the most popular graphene-based materials in medicine. Automatically, you see that you have graphene oxide coming out as the prime material of choice for the reasons I just explained to you. So the first thing we did is we invested a lot of time, and you see all of these papers that are kind of reflecting this effort, in order to identify and establish a clinically, what I call a clinical grade, a medical grade graphene oxide material. What do I mean by that? Not only to comply these around the bracket of properties that you see here, so extremely thin material, very well surface characterized, but also to have this kind of batch-to-batch reliability and reproducibility. And we are making sure that every time we make a new batch of materials in the laboratory, or if we upscale, we always maintain this set of parameters. The second thing we try to do is vary one parameter only. And this is what we've done here. You can see atomic force microscopy images of the 3 generations and 3 types of graphene oxide materials we established in the lab, and which were the ones that we would push through the clinical pipeline. These we call large graphene oxides, which are in the micron scale. The sheets, the flakes of graphene oxide materials are in the micron scale. What we call small graphene oxides, which are in a low hundreds of nanometers in lateral dimension, and the ultra-small graphene oxides, which are in the dozens of nanometers in lateral dimension. So we have almost 1 order of magnitude difference between the 3 types of materials. Everything else the same. The oxygen groups on the surface of these materials, of these sheets, are the same. The thickness of the material is the same. The fabrication is the same. Impurities are the same. Exactly the same. Also very important to note is the lack of endotoxin impurities. If you have endotoxin impurities in your material, you will carry them through in whatever biological studies you decide to undertake. So that's another thing we were very, very meticulous about. The second thing we invested The second thing we invested time, sorry, I'm revealing all my secrets. Sorry. The second thing we invested time and resource on was to go through a very thorough preclinical study pipeline. So we pushed the materials through in terms of identifying our limitations using animal work. A typical study platform is shown here. So we had our graphene oxide materials. In this particular case, I'm showing you one example. We compared large graphene oxides and ultra small, so the extremes of our lateral dimension variability. And we compared that to multi-walled carbon nanotubes, the type that was carcinogenic, the type that the regulators didn't like. So that was, if you wish, from a toxicological safety point of view, our positive control. And we compared everything else against those multi-walled nanotubes. And what we've done, just to give you again a flavor, is intratracheal, intranasal, and other installations in the pulmonary system of those animals, and we assessed safety in the context of pulmonary reactivity, inflammation, et cetera, et cetera. So lots of different studies that you can find in the literature here if you're interested. The other point I want to stress here is that you can do, which we did, single pulmonary exposures and multiple repeated pulmonary exposures, collected tissues and assessed the animals and how they responded. The whole remit behind this work is to identify which materials are giving us, are flagging up safety concerns or safety risks. And you can see I'm going to have only 2 data sets here, just for you to get a flavor of this type of work. We first assessed histological analysis after multiple exposures shown here. So if you look at the bottom panel, our multi-walled nanotubes, the bad type, and this is 1 day, 7 days, 28 days, and 84 days. So you see long-term assessment of the exposure from installations of multiple exposures of these materials and multiple doses. And you see very clearly here that the reaction of the lung tissue is not what you want. Okay. is not what you want. So you have, again, things flaring up, and you have fibrosis building, and you will have all these hallmarks of cytotoxicity, as we know, and it's very well established in the literature. Now, if you look at the control, which is the vehicle alone, you get very little response, and that's where you've framed the mapping of your system. So you look at the ultra-small graphene oxide materials, you have a very mild inflammatory response to begin with, which, as I'll show you in the next panel, is responsible, and you want that because you want the material to be cleared by the lungs. And then you have the large graphene oxide materials where you start having, at the longer time points, some time of a remnant of cytotoxic responses, and this is where high-risk material is kind of flared up. So the other data set I want to show you is how does the material interact with the tissue and the cells, and whether the material is cleared and whether it's picked up. So you see at the top panel here, ultra-small graphene oxide and large graphene oxide sheets from the same exposures that I've shown you here. And these are sections, tissue sections of the lungs of those animals at different time points, and then Raman mapping in order to identify if the material is still in the tissue or whether it's cleared. And you'll see very clearly that the ultra-small material, after about a week, you have a very few material remaining in the tissue, and after a month, everything is back to baseline. Whereas again, with a large GO, you start having some kind of deposition of the material in the long term. And at the bottom here is pretty much similar data, but instead of doing sections of the tissues, we are doing bronchoalveolar lavage. So we're taking the fluid from the lungs and we're assessing the cells that are in the fluid of the lungs. And again, we can see the presence of the materials, of the large graphene oxide materials, for long periods of time. So we decided that if we're going to run a clinical program, we should exclude large, micron-sized graphene oxide flakes from all of our human exposure studies. And that's what I'm going to show you now. I'm going to show you two data sets with two different clinical trials we've run, one for safety and another one for medical device development. But both cases of clinical translation will evolve around using graphene oxide materials. So let me start with the first one. This was a study that was funded by the EPSRC in the UK and the British Heart Foundation. And we decided to be courageous and run this study as the first in human ever controlled exposure study using healthy volunteers. The whole purpose behind the study is to de-risk the material. Remember, this is a material that a lot of people are not familiar with. The regulators are not comfortable at all. They are very cautious. They don't know what the material is doing. And we wanted to de-risk the whole landscape. So the study we decided to run was with a collaboration with the University of Edinburgh and the Dutch National Public Health Institute. Why? Because they were possessing this exposure chamber. So this chamber was transported from the Netherlands to the hospital in Edinburgh, and is basically consisting of this exercise bike setup. So the volunteer sits there for 2 hours, is biking, and is wearing a mask, and is inhaling all kinds of possible pollutants. You may freak out as nanomedicine researchers right now and tell me, what the hell are these guys doing? But from a public health researcher point of view, this could be considered standard practice in the sense of clinical assessment. For example, a lot of studies that you see already published in the literature assessing the impact of diesel exhaust nanoparticles are using this type of exposure chambers. So we decided to follow a protocol which is very comfortable to the regulators, the ethicists, and the public health officials, and instead of exposing the healthy volunteers during bicycle exercises for 2 hours, instead of exposing them to diesel nanoparticles, expose them to graphene oxides. I do not have time to explain to you the details of the study, But the paper will be published very, very soon, so I corrected the galley proof last week, so you will be able to read it at your own time. I just want to highlight a couple of points in the design of the clinical trial. We're talking about 3 visits that our volunteers undertook. We're talking about an investigator-led trial, so it wasn't a trial that any company kind of organized or funded. We're talking about 2 hours inhalation exposure under cycling, as I mentioned, and actually the difficult task was to get the volunteers come back into the clinic at repeated exposures and maintain their health status, because you want to make sure that they are not having any pulmonary infection or other disease, as you can imagine. And we are looking, the whole study was looking at the acute cardiopulmonary endpoints. It was a single site at the University of Edinburgh, a single dose, 200 micrograms per cubic meter, which I can explain to you what does that mean. Basically, we tried to determine from the published work what would be the exposure levels of workers in a factory fabricating graphene and graphene oxides, and I think quadrupled the dose to be on the safe, to be at the upper limit of what would a possible exposure undertake. And we had 14 healthy volunteers that enrolled. So I'm going to run through the data relatively fast for the interest of time. I just want to highlight again that the volunteers were exposed with either small graphene oxides, which are in the low hundreds of nanometers in lateral dimension. You can see here the distribution. And the ultra-small graphene oxides, which you see are much tighter in distribution. They are in the very low dozens of nanometers of lateral dimension. So first, what we assessed was their lung function and their heart function. And what you see in these graphs in between those 2 dotted lines is the physiological ranges you would expect. And as you can see, in both lung and heart function, both in terms of capacity and in terms of pressure, blood pressure, you see that we are not we're not triggering any accelerated heart rates or any difficulty in breathing. Again, we're talking about repeated exposures after 3 visits, but acute assessment. The other thing we looked at was the systemic plasma inflammation by looking at cells, the number of cells, and a few key markers. There are many more in the paper, but I'm just highlighting the usual suspects here, TNF-alpha, IL-6, and CRP. And you'll see again that we have almost physiological levels after the repeated exposures. And we decided to go a little bit deeper into the proteomic analysis of these patients' blood samples. And we see again that we have very few changes. So you see here all these dots are the differentially abundant proteins that are either upregulated or downregulated post-exposure, and you'll see very few proteins that are either upregulated or downregulated compared to controls. So the conclusion here is, again, subtle changes in very small number of plasma proteins. All of this suggested that, first, our thin medical-grade graphene oxide nanosheets that are in the tens and hundreds of nanometers in lateral dimension did not lead to an acute cardiopulmonary response in healthy volunteers. The second, that the same material that was intended for pulmonary exposure did not present with any hallmarks of systemic inflammation, which was also very important to see if we trigger any systemic inflammation from acute exposure. And of course, I have to say that the study has its own fundamental limitations as the first in human study that has ever been reported for exposure, controlled exposure of graphene oxide, in the sense that we are not looking at long-term effects from the exposure. We are not monitoring the patients, the volunteers for years. And that was a single dose study. We didn't dose escalate. So it wouldn't, if you ask me the question, that is not a pharmaceutical toxicology study or a medical toxicology study will always entail dose escalation. We did not do that in this study. So I will leave you with that. If you have any questions, I'm very happy to discuss. You can read the paper very soon, this week or next week, that's going to be published. And the news is kind of positive. I don't know if it's positive or negative. At least we didn't find any acute. But remember, the whole philosophy behind this work was to de-risk the material. It's not to prove that the material is safe and everybody can start inhaling graphene oxides, of course, left and right. In parallel to this clinical study, we had another clinical program that is still ongoing, and it's taking place with a very different design and very different purpose. Here, we're looking at graphene-based neural interface devices, and the whole idea is very different here. And the philosophy of the clinical development program is very different here, because here we want to demonstrate advanced material proof of concept function and safety, not only de-risk the material from a pulmonary safety point of view. This started through the Graphene Flagship Project, which was the largest ever project that the European Commission funded. And when we were putting this program together, we wanted to identify first that we are able to get unique properties that graphene materials are offering, particularly combinations of proteins, of properties, like the ones I'm showing here: electrical mobility, flexibility, transparency, and heat dissipation. Build devices that are harvesting all of these properties simultaneously, and try to assess different types of devices and applications that we could, again, push through the clinic. We decided to focus the program around brain sciences for two reasons. One, because I found it personally exciting, and I thought we lack technologies in order to investigate the brain and its functions. And B, because it represents with an array of different pathophysiological and clinical needs that I don't need to educate you. I think you know about the aging population, neurodegenerative diseases, et cetera, et cetera. So what we've done is basically summarized in this graphical illustration here. We wanted to design graphene-based devices, shown here, that will be implanted in the brain of patients and will be able to allow us multifunctionality, in the sense that we want to record electrical activity from the brain. We want to simultaneously have a device that will be able to stimulate the brain, so put electrical current into the brain for therapeutic purposes. We want to make sure that we have biocompatibility all along this process. We want to have very flexible substrates in order to facilitate surgical, minute surgical implantation, and we want to have the capacity also to build in a therapeutic delivery element. I will not show you all of this. It's impossible to show you all of this. I will just show you which of the iterations we decided, and we were able to push forward into the first in human of this clinical device. First, the science. Again, as history brings it, the paper has been published last week after many, many years of efforts, I have to say, which is not reflected in the submission and acceptance dates of the paper, I have to say. So what we've done here, the starting point is graphene oxide suspensions, similar to the ones I showed you before. Here, we're not using it as a suspendable material that the patients inhaled in the previous study. Here, we are fabricating an electrode. So we are taking this suspension of graphene oxide, we're pushing it through a porous membrane, we let the graphene oxide form a film, a membrane, and then we're transferring this using 4-inch wafer technology, I'll show you in the next slide, into a substrate that will be electroconductive. And then we're taking this, we're baking it, and we're converting graphene oxide to reduced graphene oxide, because if you use graphene oxide, you wouldn't be having electrical conductivity at all, because it compromises electrical conductivity. So we want to use reduced graphene oxide electrodes. That's a paper, you can find it online, called Nanoporous Graphene-Based Thin Film Microelectrodes, and I'll show you very quickly how we developed this into the clinic. First, let me show you how these things look like. These are the type of flexible devices we are generating, and we've done designs for epicortical, so surface of the brain implantations, or deep structure, what we call intracortical flexible devices, which are shown here. And you see those black dots, these are exactly the dots that are consisting of graphene. Everything else is based on pretty much gold and a polymer substrate. In most of our cases, we use polyimide. We've tried different other polymers, but the clinical product is based on polyimide. The second technology development we followed in this whole program was to use 4-inch wafer technologies that will allow us to design reproducibly those devices and increase the number of contacts. The more contacts, the more fidelity of the signal we will be recording. So I will show you just one example from the paper for the interest of time again. This is where we're placing the device in the auditory cortex, and we are recording when there's a sound. We're recording the activity of the brain at the auditory cortex using the device, and we're comparing it again to the benchmark. If you use the NeuroNexus benchmark, this is how the devices we used in this study look like. So you see the graphene is these black dots. Everything else is gold and a polyimide substrate. And the NeuroNexus device, which is the preclinical, not clinical benchmark, is based on metals, platinum, iridium. And the bottom line of this, for those of you that are not experts in that space, is that you have some key performance indicators. 2 of those are shown here. One is impedance. The lower the impedance, the better the performance of your device. And you see how the 2 compare. I have to stress that what we call ignite, which is the graphene contacts, are of much smaller diameter than the platinum iridium. And the other one is the signal-to-noise ratio, and you see that you're miniaturizing and at the same time maintaining or improving a little bit the signal-to-noise ratio. That means that you're able to convert a lot of activity, record activity at higher fidelity without compromising your signal-to-noise ratio. Bottom line from all of this work that's summarized in those papers, what we've done throughout this work is to basically expand the bandwidth by which you can record brain activity using graphene at both ends of the spectrum. The high end, using this Ignite microelectrodes, but also using transistors with very low frequency recordings. So basically, what we've done in terms of brain activity recording is to be able to record. Now, what would that be of clinical use? I honestly have no idea. And more concerning is that all of my electrophysiology and neuroscientist colleagues have either no idea. So we are able now to record activity from our brains at frequencies that have not been possible before, but without any neuroelectrophysiological significance. And again, that generates a question about artificial intelligence and who owns these recordings and what do those recordings mean, et cetera, et cetera. So very fun stuff to think about during the coffee break. I'm going to spend very few minutes about how we clinically translated this. And I think what I'm going to say resonates a lot with what Fran has mentioned. We were academics. I mean, I'm actually not in the business of spinning out companies. I was very resisting to spinning out companies for a variety of reasons I don't want to tell you right now. But the bottom line was that as you're developing this program and you start generating these technologies, even when you talk to pharmaceutical companies or medical device companies, they tell you, we don't want to talk with you guys. You're from Institute. You're from the University of Manchester. Who gives a damn about the University of Manchester? We are not doing business. We need a commercial entity to do business with. And that's what brought us to this spin-out company, which, again, was launched in December 2019. It was privileged to raise a lot of millions from venture capitals in order to develop the devices I showed you. My task was to get some of these devices through the first-in-human and clinical pipeline. But before we do that, we needed to transition from the little mice, C57s, bulb Cs, maybe rats, to a human device during COVID. And that slide here is summarizing the problem and the challenge, the upscaling, something that we didn't even really consider. So we're talking 2,000 to 2,400 times upscaling that needed to be done. So we needed to go from these devices that we used for mice and rats to do all of our nature papers into this device that looks kind of ugly, but if you go deeper and you dig deeper, you'll see that it has all of these features that I described to you here, but it's at a scale that can be used on a human brain. And that's how the device looks like. Remember, when you're developing a clinical program, you always, or preclinical program for that matter, you always need to benchmark. Again, Fran mentioned this need that they met to benchmark against liposomes and AAV. Here, we are in a bit of an interesting position because this is the clinical benchmark. So you can see what the surgeons are using right now in the clinic. They're using 4 contacts of platinum iridium, and they stick this plastic device on the brain of the patient. They don't let it in. It's like an acute on-table procedure that they use the electrode for. But the proposition we are coming up with is to use this device, which contains this number of contacts. So each of these islands will contain about 600 to 700 of these contacts, and they will be able not only to stimulate, record at a massively improved and high fidelity recording that they don't know what it means. So basically, what I'm trying to make you think about is, are we overkilling it technologically or not? And I cannot answer this question. Don't ask me, because I have no answer to this question. I don't know if we're overkilling it or not. What we wanted to do is push the technology in the clinic and demonstrate that it can at least do equivalence to what is done with a clinical device that is shown here. And this is the state-of-the-art preclinical, not clinical control. The FDA is very excited with this technology. They gave us breakthrough designation, at least for the combinatory part of the technology, which is great because you got the ear of the regulator. The study that we're carrying out right now is in the UK, so it's under MHRA territory, regulatory-wise. I've spent many, many hours talking to the MHRA and various regulatory experts. The bottom line is that we're running this study as we speak. This is the device. This is preclinical data that I'm showing you with, but it's exactly how the device is going to be used in the patient, in the human patients. This is done in the context of a glioblastoma resection setting. The way in which these devices are used are to distinguish between the tumor tissue and the normal brain tissue. The normal brain tissue electrophysiology is very different to the glioblastoma tissue, and this is again an on-table. It's going to be used like that, so it's only surface recording and stimulation of in order to distinguish and run very highly precise distinction between the tumor tissue and the normal brain tissue, so that they will be able to resect in a more precise manner. This will be a class 3 brain monitoring and stimulation device. Just some details about the study. This is a prospective, interventional, non-randomized, single-arm, single-center study. It's taking place as we speak at Salford Royal outside Manchester, and we have 8 to 10 adult patients with a suspected glioma that are planned for surgical resection, and we will follow those patients up for 6 weeks. The whole study is very simple in a way, in the sense that these patients will be undertaking resection of their tumors. All we have been granted, and we've requested regulatory approval for, is to open up a bracket for half an hour and substitute the clinical device I showed you in the previous slide with this graphene device, perform exactly the same recording and monitoring, distinguish tumor to brain tissue, and then, so it's a plug and play. Take out the graphene, put back the electrodes, and complete the study. That will be the first in-human study for any graphene device to be completed in 2024. If anybody knows of any other graphene device that has been tested in any clinical, but in contact with the brain, there's lots of graphene biosensing devices that people put blood, saliva, whatever, urine on it. But this is the first interventional product that I think we are pushing out. Okay, I completed my I don't know if you want to run through some lessons from the clinical translation, maybe not. I will just run through very quickly the 4 lessons, and I want to highlight on one, which is this one. The decision to the optimum time to a first-in-human clinical trial. This is a critical decision that I think could be catastrophic potentially to a technology. If you rush very quickly, you may prompt the decision oh, this technology doesn't work, dump it. But if you delay too long, you may run the problem that Fran alluded to, which is you're running out of money. You're running short of money and resource. Or the technology loses excitement from the community. So it's like, oh, established kind of thing, doesn't really do what it's supposed to do, let's dump it. So again, you need to tune and find out and be very lucky with the way in which the rest of the landscape is morphing. For example, in these neural interface devices, we were very lucky that the people like Elon Musk and crazy other people are trying to put a lot of investment and explore this area with companies that want people to connect the brains with the internet or other crazy silliness that I hear. But on the other hand, they created a very positive landscape for the development and the reception of this technology from a funding and investment point of view. I will leave it at that, and I have only two things to say, to thank the team. So we had our Nanomedicine Lab for many, many years, almost 30 years. Beppe reminded me this morning. And now it's translating into independent laboratories. So we have spun out all of these laboratories from the Nanomedicine Lab. 3 of those were contributing to the program and the data I showed you. Cyril Boussy's Nanoinflammation Lab contributed to all of the preclinical work I showed you with mice and the pulmonary inhalation, including the first-in-human safety study. Marilena's Hadjidimitriou Lab, Nano-omics, contributed with the proteomic analysis of the blood samples using our nanoparticle technology. And of course, the lab in Barcelona at the ICN2, which has been responsible for the fabrication of these clinical-grade graphene oxide suspensions, has contributed a lot. If you're interested still about what we do in our laboratories, Sandra, who is here, I think she's at the back, she will give you 2 oral presentations during the meeting this afternoon and tomorrow. And if you want to listen to what is closer to my heart from all of this, which is liposome research, you are very welcome to listen to our PhD student, Noelia, who's going to give a presentation on Wednesday afternoon. Thank you very much. These are our collaborators, and I'm sure Beppe wants to get rid of me here. Thank you.
Thank you. Brilliant, brilliant talk. Questions? We got about 5-6 minutes for questions, and perhaps we can sacrifice some of the coffee break too. Anyone? Yeah.
Hi, thank you for this beautiful talk. Congratulations for the huge milestones. So I was wondering, when it comes to the clinical part, using like a new device, Graphene, etc., does it require training the medical staff to use this and also interpreting the results? Or do you plan, for example, to prepare software to distinguish between healthy and tumor tissue? Thank you.
Thank you. As in the context of the clinical trial, it does not require any training, other than the fact that you have scientists from the company, InBrain, working with the clinical electrophysiologist, because the whole remit of the clinical trial that I showed you is equivalence. You're not trying to demonstrate that you are performing over and above the established technology. That's the primary clinical endpoint. There is another endpoint, which we are not talking about, which is actually to get the data, because we're going to get the data anyway. So that becomes more of a clinical research project. And we will be able to try algorithmically to understand what are the electrophysiological signals we're recording with those devices compared to the metallic clinical control. And that will be done primarily through the signal analysis group in the company. In the long term, the technology is, as I mentioned to you, the success of the technology is based on this capability, the expansion of the electrophysiological signals you're able to record. Now, what will that mean is a different discussion that I cannot really, nobody knows. It's like designing a new hydrophone, for example, for the ocean, and you hear things in frequencies and you hear whales and dolphins talking. Do you know what they're saying? No, you just hear clicking noises, and so you don't know exactly what's going on. So that's the similarity. That's a parallel in which I kind of can offer.
There's one question there.
Hi, thank you so much for the really interesting talk. So I have a question about the part where you did the de-risk study.
Yeah.
So you did it on people through inhalation, and I thought it was interesting. I haven't really come across that before. So is there a reason for why you chose that method for the exposure instead of, for example, injection? And I was wondering if inhalation is the method that you were thinking of for the administration in the future?
Mm-hmm. That's a good, maybe for the non-nanotech experts, that's a very good question. The whole of the nanotechnology, nanotoxicology space has been dominated by pulmonary safety studies. So if you look at the studies from carbon nanotube, for example, why carbon nanotubes, if I ask you here, I'm sure you're going to tell me, oh, it's toxic. Why is it toxic? What is the toxicity based on? There's been no clinical study with carbon nanotubes injected. There's only one study whereby there is a Nature Nanotech study of 2008 by our colleagues in Edinburgh, by the way. Half of them are retired now, which they exposed rats into pulmonary-wise with the same nanotubes I showed you, and then asbestos fibers. And they correlated, and they saw that there is the pathophysiology in which the tissue is responding is very similar. However, if you take these multi-volt nanotubes, you chop them up, you lose that. Why haven't nanotubes been developed further in the clinic? I don't know. People think it's toxic. So I'm just giving you one example of why pulmonary toxicity has been a hallmark in assessing nanomaterial toxicity and safety. And that's one of the drivers. The second driver is what I mentioned to you while I was presenting the design of the clinical study, which is we tried to base this first in human. Remember, there's been no exposure of a human being on graphene oxide. Where do we start? With a setting which will be acceptable to the public health officials and the public health authorities. Because the first thing is not medical application that they're going to be concerned, but passive exposure from materials. And the first place in which this is going to occur is the factory which produces these materials. That's why we went to the pulmonary exposure.
Thank you.
Okay, a question here and then Sundar.
Sorry. Go ahead. Thanks. It's also related to the previous question, and it's basically about, you say this is going to be in the industry, basically, where the workers are going to be exposed to this graphene oxide. So it's not already like in process of producing, right? Or it's already producing?
There are companies that are producing maybe ton volumes of graphene oxides at the moment.
So there's a way to measure the graphene oxide in the industry's area?
Yes. So if you remember, the dose that I showed you is 200 microgram per cubic meter.
Yes.
This is the exposed dose in our study. The studies that we saw in the literature and the ones we based our exposure level that are reported is 0.5 to 4 micrograms.
Okay.
Okay. So there's one study that is reporting 40 micrograms per cubic meter exposure. So you see, so we took the highest. There's only one study. Most of the studies are 0.5 to 5. There's one study that reports 40. And what we did is we said, okay, let's do 5 times this.
Okay, that was basically part of the thing.
So that's how we kind of, because we only had one chance. As I told you, it was only a single dose study. We were not allowed to dose escalate.
Yeah, and also the second question, sorry, is that with this small graphene oxide that is entering and being excreted by the body, do you know how it's excreted, and if it's possible to use this small graphene oxide as a possible absorbent for other nanoparticles in our body in order to...
Yes, the second I would say no. It's getting too complicated for me to answer, so I wouldn't use it as a platform to absorb other things, even though, having said that, there is a product in the market which is very funny, I think, which is called carbon black. So if you have some kind of I don't know if anybody has seen that you can drink a carbon black. I don't know how your urine will look like after this, but anyway. So the concept is there that you use carbon in order to absorb, I don't know,
Nanoplastics, everything, yeah.
impurities in your. It's a little bit Trumpian, this kind of approach for me.
Yeah.
But anyway, the first question you asked was about clearance mechanism. We know that macrophages and neutrophils are very quickly recruited in order to capture the material. Now, another question is what happens after capture? This is a little bit more difficult to assess because there are 3 processes taking place at the same time, which we have published, and you can find it in the papers. 1, degradation intracellularly. 2, clearance from the tissue and localization into the lymphatic nodes, where probably they stay longer and get degraded. 3, clearance from the lung into systemic circulation and excretion. So it depends very much on what type of material you have and what is the surface properties of these materials in order to determine which of the 3 processes, post-exposure, post-localization, will be predominant. But all 3 are taking place.
Okay, thank you.
Just one question there, very quick, please.
Thank you for your talk. I have a question. I noticed you do the inhalation to our lung. So
wait, you are too quick.
Oh, sorry.
Go.
Sorry. Yeah, I noticed you do the inhalation to the lung.
Inhalation to the lung.
And as we all know, the graphene is difficult to degrade in our body. So with our time go.
I didn't say it's too difficult to degrade.
Actually, before we do the graphene, we notice it will degrade at least 4 days. Okay. Yeah, we do at a 4 days term. We notice when we do inhalation, the graphene will accumulate in the lung over time. So it means the inflammation response will also increase. But I notice your article only records 6 hours. So I want to know how to prevent the long-term inflammation and the immunogenicity, and how about the long-term organ toxicity. And the last question is, how do we know the safe dose of the graphene? Yeah, if we can control safe dose. Yeah.
Can I answer or no? Well, it's up to him.
If you have a quick answer,
Okay.
yes.
I do not have
short
a quick.
version,
I do not have a quick answer to your. You're asking very fundamental questions here. So, but they have a lot to do with the question that the gentleman asked me earlier. First of all, the degradation is taking place. So that's very clear. The question is not whether degradation is taking place. The material is degradable. The key question is, what are the kinetics of degradation? Okay? Degradation is taking place. But if it's taking place in a decade, and you keep getting exposure faster than your degradation, then it's a kinetic problem. It's not whether degradation exists or not. Let me clarify this. The more oxygenated and thin your graphene oxide is, the better and faster it gets degraded. So it will depend very much on the type of graphene oxide you're performing your studies with in order to have degradation kinetics that will beat exposure kinetics, okay? So therefore, I cannot give you an overgeneralized question, oh, graphene oxide is safe or not safe. It depends very much what you want to do, what is your exposure levels, and what is your kinetic of degradation levels. That, I think, we can take forward for coffee if you want to counter-argue with me. With regards to safety of the material, I am not able to give you a blinded answer for any material, including water, whether it's safe or not, unless you tell me how you're going to use it. If you take water and you start injecting yourself with water, you're going to die at some point. Is water toxic? I don't know. So these kind of questions I cannot answer, and I've been asked about them for 35 years. I cannot really answer these questions unless they are framed in a very specific profile. So in order to give you a fair answer to your question, I would say, yes, it's toxic if it gives you a massive inflammatory reaction that you wouldn't be able to control, so it will be an acute response to exposure, but it wouldn't be that risky if you have a measured inflammatory response that you kind of need in order to clear the material from the tissue of exposure. Because inflammation is not only bad, It's also good if you want materials to clear.
Okay, I really like this. So we're going to take one last question.
Oh.
Because Kostas is getting angry. It's a start to
This
the
is-
right way.
you didn't even pay me for this.
You were dedicated and vocational.
Okay, let's go.
There, in front of
Hi.
front row.
I have a question also. Well, first of all, thanks for
Not
your
about
presentation.
safety, please.
No, not about
Okay.
safety this time. On the first part of your presentation also, like on the water dispersible graphene oxide nanosheets, I understand that the study was mostly for like passive exposure, but do you have any applications, like biomedical applications envisioned for that kind of materials?
Mm-hmm.
What's the most promising in your opinion?
Thank you. That's a good, creative, constructive question that I'd love to answer. Yes, is the answer. And if you look at the literature, and what we try to publish is to develop graphene oxide suspensions as platforms for presentation of biomolecules. And this can be for immune triggering processes, and whether you can modulate the immune system, because the advantage you have is the very high surface area. So you can do multiple presentations of biomolecules in this platform system with very little effort. If I compare to, say, liposomes or polymersomes, or where you need to encapsulate really the material and control the formulation. Here, you can absorb biomolecules very, very quickly and very, very easily. Now, whether this will be as efficacious or not, it has to be framed in a very specific context. But that's the kind of more creative, if you wish, medical application we're exploring.
Of course, thank you.
Okay, thank you very much. Please join me to thank Kostas for his talk.